Marilla Gennaro talking about biomarker core and then machine learning from Princeton. Marilla, you want to take it away?
Yes, yes, let me just see. Do you see my screen?
Yep.
Do you still see my screen?
We still see it.
Okay, it's a Mac.
No, it isn't. No, no, that's a PC.
No, it's a Mac.
Mac is a,
Okay, so good afternoon, everybody, and thank you for still being here. So I'm going to talk about our biomarker core. So I don't know exactly what team science is, but I think I practice it. So that so here is the group of in my lab, the names of the people in my lab, and on the right side, let me just close this, uh, are uh, Rutgers collaborators. This list, with the exception of Ray and Alfred Lardizabal, that are both pulmonologists, everybody else has become a collaborator since the beginning of the COVID pandemic. And this list is even not complete, and it grows by, I don't want to say by the day, but certainly by the month. Um, okay, see now to give. Okay, so I want to start with where we are and then take you how we got here. So these are the projects that the core is supporting. The first one is a biomarker for asthma. This is a project that we're working on together with Ray, and it's been put on a hiatus due to the COVID pandemic. The second one, despite the fact that it's not COVID, is a TB clinical trial sponsored by Biomarker. We were able to keep it going throughout the pandemics, and actually we finished it. We're gonna complete it officially next week, and it's about testing. It's a clinical trial. The data is being done all over in several sites over the world. We've been the most active site in the US because we beat the pandemic to run this trial, and it's about a new platform for immunodiagnosis of tuberculosis. All other projects are COVID-19 related. The first one is, of course, the Rutgers Corona cohort that all of you know about. So I'm gonna tell you just a little bit about others. So we have a study on Rutgers employees who've had COVID. So we study immune responses in convalescent plasma and also cellular responses. We have a study that is sponsored by Pfizer on seroprevalence of SARS-CoV-2 infection in Essex County. And then we have a study on convalescent plasma therapy for COVID-19. And then we have a series of studies on vulnerable populations to COVID that include firefighters, children, dentists, people living with HIV, and people that belong to ethnicities that have been particularly hit by COVID. So let me tell you so that's where we are. We started in a kind of a different way. So the core was included in the hub because we were running an assay that was novel. We had developed it at PHRI, and it's a RNA flow cytometry platform that so combines conventional flow cytometry with in situ hybridization for nucleic acids, and is particularly suited to study stages of disease and risk of reactivation, exacerbation, so dynamic states, because since it measures mRNA, yields much more dynamic readouts than conventional flow cytometry. And we initially started applying it or the plan was to apply it to asthma and tuberculosis. And then, as we were doing this work, COVID hit. And since I had had experience in studying antibody responses to tuberculosis, we thought that our way that we could contribute to the work on COVID was to set up a facility and use the core and our abilities to study antibody responses to SARS-CoV-2 infection. We were very lucky to be able to receive antigens produced in-house by a Pinter lab that leveraged his expertise in HIV proteins to produce quite rapidly a couple of SARS-CoV-2 proteins. Other proteins we obtained commercially. And with this a little armamentarium and a lot of goodwill, we took off. And actually we started flying in this new era without really having a plane. And we built the plane as we were flying. And the plane is here now. So now, we started with a situation in which we had to borrow people from other laboratories. Marty Blazer very generously, and even more generously Dan Fine at the dental school, who does not, did not participate in the Rutgers Corona cohort, lended us, quote unquote, people to help us out to get started. And now we have, at the beginning, we didn't even have enough multipipeters for all these people to do the work. And now we have a BSL-2 plus laboratory that has a robotic Hamilton liquid dispenser and all the instrumentation for automated ELISA. So with this, we've tasted more than 5,000 sera, and I think the number is probably a much greater than that. As we were studying the antibody response, we realized that studying only or detecting only the presence of antibodies is not sufficient. Antibodies are there to do things. And so there are good antibodies, bad antibodies. So we set up a number of functional assays for antibodies. The first one clearly is a virus neutralization assay that we do using live SARS-CoV-2 virus, leveraging our BSL-3 facilities that were built mostly for tuberculosis work, and also our expertise to work in such facilities. We've also set up assays for antibody-dependent enhancement and endocytosis, and we're going kind of down this list, and we have reagents to set up the rest of these assays. So this part of this is in progress, work in progress. We also realize that even though there are very fine immunologists across our campus, our campuses, nobody was really focusing on immunophenotyping of memory B cells and plasma blasts, that of course are the cells that produce the antibodies. So we thought that we had to fill that gap, and I think that the core can fill that gap for the entire university. And this is, for example, the panel that we have developed to study memory B cell in COVID or in any other condition. We also have, we're going back to the fish flow, the RNA flow cytometry that I mentioned at the beginning, and apply it to COVID-19. We have the reagents to do that. So what are in summary our accomplishments? So I was asked to think about which other cores we have interacted with. Well, of course, we've interacted with Bird, with Jason Ray and his team. As I was hearing people talk, I think we have interacted actually with more. We've also interacted with David Foran's group during this work. we have done for tuberculosis work collaborations with investigators at others CTSA hubs. Um, for the COVID work, we have raised the funds through administrative supplements to NRO1, and this is for convalescent plasma work. Together with the School of Public Health, we have Pfizer funds for zero prevalence. We've been funded by several intramural grants from investigators that have asked us to run this in the assays that I mentioned, and of course we have been participants since day one in the Rutgers Corona cohort study. We have NIH grants that are pending. 4 of them, to my last count. We've had quite a few. I'm not gonna tell you how many they were submitted but didn't get funded, but they were certainly also worked out, and these are the publications that we've had most recently. And as I was looking at them, I realized that Ray is a co-author in 4 of 5 of these papers. So that's where we are. We plan ahead to be more of a core service that offers not only collaboration, as we've done so far, but also consultation in planning experiments and identifying services that are not within our area of immediate expertise or that we run, and to assist with generating data and for paper writing, grant preparation, etc. And I already mentioned the type of assays that we can directly do as collaborative work or as a service. Our future plans include establishing a website that informs Rutgers about what the biomarker core can provide. And actually, we're quite, this project is quite advanced, and there is a draft website that has not been launched yet, but we're almost there. To continue and to expand the COVID-19 related services, and also to support studies that are on different infectious or non-infectious diseases, and to increase the interactions that we've had with other NJAX cores, especially in terms of data analysis, given the fact that there are different kind of data that need to be put together, both clinical and laboratory, and so different quantitative, non-quantitative, they need to be put together. So that becomes a really important task for translational work. And I want to say at the end that all this work has been done by a core of 1 person, and that person is not me. It's Natalie Bruners in my lab, who's run and be taken responsibility for all these activities really tirelessly. And she's been helped by other members of my laboratory. And I really want to, as Ray said at the beginning, in general, I want to spend a few seconds saying how extraordinary the effort of these people has been since the beginning of the pandemic. They have not taken a day off. They have not given themselves the opportunity to get bored watching Netflix. They have been at work every day, and they continue to do this work in really an extraordinary fashion. Thank you.
Great, Marilla, very well done, and an exciting core. This core has evolved from what it was initially, a biomarkers core, but because of an unmet need and a direction and fantastic immunology, it's being now built as an immunophenotyping core right now, with a focus on COVID for obvious reasons, but with expansion into other disease states. Bob raised a very interesting question. How did you build and how do you support the biorepository? I can say honestly, on a wish and a dream, these things we did not in our original application write in a biorepository, and that's part of the theology and philosophy. You can start to save samples, but without a purpose, those samples often don't get used. And that's an enormous lift cost wise if you have a freezer farm, and then you have to monitor the freezers. And then you also have to be sure what your freezing still has good quality, and what's your QA, what's your QI. It's an entire science in biobanking. I was experienced in the failures at Penn. So we didn't build it until COVID came, and we then had to figure out where to put 27,000 samples. Certainly my -70s were all filled. So what we did is we actually leveraged a world-renowned biobank that was on campus at Rutgers. That is RUCDR. They were actually the first to get the EUA for a saliva test for COVID. And they literally have millions and millions of samples, all done robotically. And what we did is we leveraged that and we buy space there. But that was only a piece of it. And that's actually a large piece, the hardware. What was necessary then is to develop an honest broker system, because these samples have to be de-identified from the investigators. They have to be saved properly, QA, QI done, and then robotically and all barcoded sent out to other investigators, requiring an honest broker, not only for biospecimens, but also for the data that's associated with the biospecimens. So we all did this pretty quickly, learned a lot, and we now have, you know, a biobank that is available. We have now opportunistic sample curation and acquisition for all the patients that we can get in hospitals with COVID, and we'll make this resource available. Obviously, it'll be a cost to investigators, and we have to have a governance and determine that the studies warrant sample use. But Bob, we didn't go in this eyes open saying we're going to build a biorepository. I could assure you, if COVID didn't come, we would still be wondering, how are we going to build a biorepository? So, you know, you want to talk about the good, the bad, and ugly. I wish I could have told you this was unbelievable foresight. No, it was dumb luck and need. So that's the way it worked. All right, let's move on to machine learning. This is the next core.